Good morning, everyone, and welcome to day 4 of the Kernza meeting. My name is Tessa Peters, and I'm the Commercialization Manager at the Land Institute. Our first presenter is Tim Cruz. He's a soil ecologist and the Director of Research at the Land Institute. His research interests focused on the biogeochemistry of soil nitrogen, carbon, and phosphorus in low to no input perennial agroecosystems. He's currently working to evaluate the ecosystem services that are enhanced when Kernza is intercropped with perennial legumes such as alfalfa. Thanks so much, Tim.
All right. Let's see. Well, hello, folks. Thank you, Tessa and Lee, for the opportunity to present today. All right, I'm going to dive right in. Recapturing. The soils that feed us today were all formed under diverse perennial vegetation. And soil carbon accumulation is a significant part of soil formation. It comes about by plants making investments in coarse roots, in fine roots, which release carbon-rich exudates and host mycorrhizal fungi that grow hyphae beyond the reach of fine roots and produce the sticky organic molecule glomalin that helps to bind stable soil aggregates. The totality of these organic inputs from plant roots, exudates, mycorrhizae feed an extraordinary food web of soil organisms, from bacteria and fungi to algae and nematodes to springtails and earthworms. Populations of soil creatures explode under the right conditions, and trophic groups gobble up other trophic groups, resulting in a matrix of dead and decomposing bodies, or so-called necromass. This process of soil organic matter accumulation goes on until respiration by this amazing soil food web approximately equals plant inputs from photosynthesis. And then humans came along and are the first organism to convert this perennial vegetated landscape into a status of annual plants that persists today indefinitely, especially with the addition of nitrogen fertilizers, which allows for continuous cultivation. And this conversion of perennial to annual vegetation has resulted in a retrogression of soil formation, including loss of soil organic matter. Why is soil organic matter lost? Well, when soils that were stable and were allowed to form aggregates are disturbed with the plow, it breaks open these aggregates, and physically and chemically protected forms of organic matter are made vulnerable to microbial attack. And so microbes have a heyday. They chow down on the soil organic matter and convert it back to carbon dioxide in the atmosphere. So losses of soil organic matter increase under annual tillage. But what is often not recognized is that the perennial vegetation that allocates on the order of 40 to 70% of its annual productivity below ground is converted to annual vegetation that allocates only on the order of 15 to 25% of its annual productivity below ground. So not only are carbon losses increase, but carbon inputs decrease under annual grain cultivation. And this has resulted in a very ubiquitous and widely recognized decrease in soil organic matter under annual crops compared to the ecosystem that was there before. And this figure by Schlesinger shows loss of soil carbon over six decades in 2 soil types, but these 2 soil types kind of bracket the range of soil organic matter loss that is typical in conversion to annual agriculture, somewhere between 20–25% of the carbon and 60% of the carbon. This has not gone unrecognized. In the last decade, with the interest in looking for sinks to put atmospheric carbon, a lot of folks have gotten interested in regenerative agriculture, and the core of which is really building soil organic matter. And this program, or this approach that's promoted by NRCS, shows up everywhere. It's quite remarkable. The drive to maximize continuous living roots, minimize disturbance, maximize soil cover, maximize biodiversity is promoted widely, and is also 4 of the tenets of the Soil Health Institute. And so there's good reason, based on what I just described, that replacing annual agriculture that involves annual roots and a lot of disturbance with perennials should result in an increase in soil organic carbon. And in fact, a review of meta-analyses that Brian Rumsey and I did a few years back shows this, that a range of mean increases in carbon per hectare per year ranges from 330 to about 1,900 kilograms of carbon per hectare per year across plots, CRP plots, where annual cropping systems were converted to grasslands, and also in cellulosic bioenergy crops. And so if you see numbers that are a lot higher than this, I suggest you question them. The challenge with many things we do is in the methods of measurement. It's very hard to measure soil carbon accumulation. Estimates the net carbon balance of an ecosystem, and its plus is that it's pretty reliable, and that it actually kind of averages out an entire field. The minus is that you cannot differentiate soil organic carbon at the next couple of slides. This is some work that was led by Gabriel de Oliveira, working with Nate Brunsell at KU. Gabriel is a postdoc. And these are eddy covariance data from a Kernza field here at the Land Institute. The top figure is net ecosystem carbon accumulation. How much carbon is the ecosystem gaining every year? And then the bottom figure is percent of gross ecosystem carbon uptake or photosynthesis on an annual basis lost to respiration. And so, and 2013, the starting year of this figure, is 4 years into when this field was actually planted. And so what we see is that the peak amount of carbon accumulation that happened 5 years into the planting of this field. And that number is somewhere around 5.6 tons or 5,600 kilograms per hectare per year. If you go back to that number, that's a lot higher than one would expect soil to be accumulating carbon. And so what that's reflecting is the building out of the Kernza crop in living biomass, the roots in the above ground vegetation, as well as non-decomposed residuals. But as we see over time, respiration starts to catch up with productivity, and eventually you start to reach close to an equilibrium. But if carbon is being sequestered, you're never going to get there, okay? And that's what we think we're seeing, is that we're approaching a time in this field when the eddy covariance is starting to show the rate of soil accumulation of carbon, which would probably be somewhere between 0.5 ton and 1,000 kilograms per hectare per year. Oops. Another eddy covariance tower project that Gabriel was leading took place at the Perennial Agriculture Project outside of Lawrence. We converted a restoration prairie, a pretty weedy restoration prairie field to Kernza and alfalfa biculture, and had an eddy covariance tower to witness this conversion. And we had a year of baseline data leading up to the conversion. And what we can see in this figure, this is day of the year down here on the x-axis, was September 15th being the initiation of this period of time. Over the course of this year, after 10 years, this restoration prairie was still accumulating on the order of 810 kilograms of carbon per hectare per year. That could be soil organic carbon, because 10 years is a long time to build out that prairie vegetation, and it may have reached equilibrium. And so it's starting to reflect a number that seems believable in terms of soil carbon accumulation. Then we made the conversion, and in this year, so this is the year after conversion, sure enough, we lost 1,210 kilograms of carbon per hectare per year. And that reflects both the vegetation decomposing as well as loss of probably particulate organic carbon from the disturbed soil. This was plowed in the conversion. We did that intentionally to see how much carbon would be lost with plowing. But interestingly, second year after conversion, the growth of the Kernza alfalfa intercrop had just about matched the respiration losses from the disturbance. And we're seeing almost equilibrium of gain and losses. And we suspect that in the third year, there would be a net accumulation of carbon. And I don't call it sequestration, again, because that accumulation is largely going to be in the living biomass at this point in time. The other common approach to measuring soil organic matter, and probably the most common actually, is in using soil cores and estimating the carbon in different strata, and then comparing different treatments that have had, perennial crops versus annual crops, or no-till versus tilled. And it's solid, but there are challenges. The pluses are that it's possible to estimate soil organic carbon with depth, and that soil carbon can be fractionated into meaningful pools with permanganate oxidizable carbon or mineral associated organic matter and particulate organic matter, these types of pools. The minuses are that it's very difficult to measure significant changes. Soil organic carbon is very slow to accumulate. You're adding a little bit of carbon to what is often a large pool, which is the case at least in these Midwestern mollisols. And soils are highly variable, and the sample size is incredibly small that we pull in these cores relative to the total field. If you're down in our part of the world, where we have some alkaline soils, where we don't have a lot of precipitation, in excess of evapotranspiration, we get carbonates, and those have to be eliminated because the inorganic carbon interferes with the organic carbon signal. And then also very problematic is bulk density methodologies are limited. They're very difficult to replicate in time, and especially if there's different research groups doing it. There's just it's a bit of an art and a science to measure bulk density, especially with depth, if you're going deep down in the soil. In this paper in 2014, in the Journal of Soil and Water Conservation, Nekpalova published these minimal detectable differences necessary to actually, how much time does it take to measure minimal detectable differences in soil organic carbon? And they showed differences between treatments, and estimated on the order of 8 to 100 years with 5 replicates, and tracking single treatments over time, such as a no-till field, 11 to 71 years under that treatment before you have the confidence that you can really see detectable differences in soil organic carbon. This is pretty sobering for people who are writing grants that last 3 years and they want to actually measure changes in soil organic carbon. Now, you would think, though, that in perennial agriculture with a crop like Kernza that has so much more root biomass than the annual crops that were behind these minimal detectable differences, that, well, maybe we could see differences in less time. Or maybe not. We've had mixed success. One comparison we have made recently was on some fields that Jerry Glover planted at the Land Institute, and on a very, very similar soil type to that where those eddy covariance tower data were from. And these were planted in about 2004, and there's a single species Kernza, restoration native vegetation that is a mixed species, and an annual rotation. And there were 3 block samples, so only 3, not 5 replicates. Soil cores taken at different strata, 6 different strata, and the samples from each depth were homogenized within a plot. So there were 3 of each strata that were batched, and then there were 3 replicates. And we had to eliminate the carbonates on all of these. And what we got were some really interesting apparent differences down the soil profile in annual vegetation versus intermediate wheatgrass, which is Kernza. This is really before it was very bred, and then a native vegetation that's diverse. And none of these differences are significant, but very interesting to see what appears to be an increase. They're on the way to a significant increase, at least in the surface soils, with the perennial vegetation, and how the Kernza seems to be standing out at all soil depths except the 0 to 10. And when you aggregate those down to 80 centimeters, it looks like this in terms of the total carbon accumulated over 16 years, or the status of the soil carbon over 16 years. And the difference between Kernza accumulation and the annual rotation is 1.06 tons per year if you make those differences, you consider them to be linear. So about a ton per hectare per year, and that falls in nicely into that range that Rumsey and I bracketed, and similar to where the eddy covariance tower was converging on. So that's interesting. But what about if you add some diversity to the mix? There's been a lot of work recently. This study from Cedar Creek in Minnesota by Young, working with Tillman in the famous plots, the diversity plots. And of course, this is a very sandy soil, so the amount of soil organic matter accumulating is low, and it's probably mainly particulate organic matter, not so much mineral associated organic matter. But as you can see, is that over 22 years, the amount of carbon accumulated in the high diversity plot is considerably greater, probably 30% greater than what you're seeing in the low diversity plots. And then the amount of root biomass really is significantly different across this species diversity. So root biomass does seem to be driving the inputs of roots, driving the soil carbon. At least that's what they talk about, and they go on at length to suggest that this is a complementary effect, not just a sampling effect that's driving this correlation between diversity and soil carbon accumulation. Well, we have had a little experiment ourselves where we had a series of plots of treatments with Kernza and alfalfa over 5 years at the Land Institute. And we had Kernza with no fertilizer added at low density, Kernza with fertilizer added at high density, and a Kernza alfalfa intercrop. And we had these other 2 treatments, but we only sampled these 3 in year 5 to compare with time 0. And so this is carbon accumulation over 5 years. And interestingly, what we see is that there was no significant carbon accumulation in the Kernza, either of the single species Kernza plots. However, the Kernza alfalfa intercrop did show a significant increase in soil carbon. It's interesting that the plots started with a lower mean carbon accumulation. It was totally randomized, but nevertheless, this is a significant increase. And if you divide that difference by the 5 years, it's an average of 1,320 kilograms of carbon per hectare per year being accumulated in the soil organic matter over 5 years, with 540 in the high density Kernza with fertilizer and 240 in the single species Kernza without fertilizer. So these are really interesting. And I think that one thing that may be behind this large carbon accumulation in the Kernza alfalfa is the greater root biomass that we see in those plots. In years 1 and 2, when we did these corings, the amount of root biomass that is averaged under both the rows and of alfalfa and Kernza. So this is roots under alfalfa and this is roots under Kernza. Was almost double that of 2 rows of the Kernza with and without fertilizer. And now we don't know that, in fact, we think it's unlikely that this is actually alfalfa root specifically. It's probably a lot more Kernza root growing under the alfalfa, but nevertheless, the total root biomass in this intercrop system is quite a bit greater than in the single species system. Another thing that may be going on in the intercrop system is that the alfalfa provides a different tissue quality than the grass alone. And Laura Vanderpool, a PhD student working with Francesca Cotrufo at Colorado State University, are looking into this in the intercrop plots versus single species plots at the Land Institute, in which the idea is that the alfalfa might add more of this non-structural labile carbon that leaves dead plant matter more quickly and feeds the soil microbial path more quickly and more substantially, resulting in higher microbial use efficiency, and ultimately the formation of mineral stabilized organic matter. And this is a pool of organic matter that sticks around a long time, as opposed to the more lignified tissues that you might expect to see in dead Kernza roots or above ground biomass that breaks down to finer particulates but stays as particulate organic matter. And while this forms more slowly, it disappears more quickly. So it's an interesting juxtaposition. This forms quickly, but the soil organic matter that's formed is very persistent. And we're looking forward to what Laura finds in this study. And just to finish off, we have a couple of new soil carbon collaborations. One is more with the Cotrufo Lab and NRCS folks here in Kansas, where we're sampling soils in farmers' fields that have Kernza growing in them, and trying to see if, in fact, MIR might be a new method to get at not only total carbon, but the particulate organic matter and mineral associated organic matter fractions in the soil organic matter developing under Kernza on farms. And then really importantly, we're negotiating with Stephen Ogle at Colorado State to sponsor a PhD student that you'll be looking for this advertisement soon. Someone who will then work on modeling soil carbon in Kernza across a wide range of soil types using Dasent with a long-term goal of adding Kernza to Comet Farm. These individual studies are extremely useful, like I was just presenting with the Kernza alfalfa intercrop. But the modeling at this point is the most robust way, given how few studies we've done. And of course, the field work and the modeling together are the best. And we think that we're going to improve in this quite a bit over the next 5 to 10 years. So thank you so much for listening. And it looks like I might have time for a few questions.
Thank you very much, Tim, for that talk. That was excellent. So I invite questions. If people have questions, you can go to that Q&A box at the bottom of your screen. Just click on that and type in a question, and we'll be able to ask it of Tim. It's probably the best way to proceed. We do have about 5 minutes of time here, so hopefully someone will have a question. I'll give you a little moment to get it in. Here we go. Question from Damien. Any comment on the above ground biomass?
Yeah, that's a good question. Basically, you know, we haven't really done the work to see how much the below versus above ground biomass matters, but for people who have done that work, especially in grasslands, emphasize the roots as being the most important source of soil carbon. Francesca did do work at Kernza and found that the above ground leaf litter did actually get incorporated to some extent, especially in the superficials, in the top strata of Kernza soils. It is partly based on the ecosystem. You would expect that in a, for example, deciduous forest with coarser woody root inputs and a lot of labile inputs and leaf litter, that the above ground inputs would matter more. But the party line is that below ground inputs, especially in grasslands, matter the most.
Thank you. Another question from Anna. Is it known if the root's biomass production is indirectly changing as a consequence of selection for higher seed yield and intermediate wheatgrass? And may there thereby decrease soil carbon accumulation in fields with the newer grain varieties?
No, it is not known. Very, very difficult to measure, especially across different soil types. I can't tell you how much soil texture matters in terms of below ground allocation. And so it's really hard to make these generalizations. But one thing we saw in Christine Spunger's figure that I put up was just how much below ground allocation there is. And I think we'd probably be willing to sacrifice half a ton to a ton of below ground allocation for a comparable amount of seed yield. I don't know, Lee, would you sacrifice that? I think, yeah, okay.
Yeah.
I mean, it's so overbuilt, and we think that that may be a vestige of below ground competition in grassland ecosystems. I mean, you know, soil carbon is huge plus here, of course. But my inclination would be to maybe reduce the Kernza root some and throw in some legumes, and I bet you're going to get more total carbon accumulation than you would have just with single species Kernza.
One more question from Jess. There are several efforts to explore legumes that are compatible with Kernza. Should we be focusing on those that have extensive roots like alfalfa?
Yeah, I think so. I think the complementarity aspect of the legume intercrop is very important. Brandon Schlautman here at the Land Institute is focusing some on Cura clover and sainfoin, and we're very interested to see how compatible those roots are with Kernza. The plus with alfalfa is that it does appear to be getting its water from a different part of the soil profile than the Kernza, deeper, that is. Kernza, with all of those roots, is getting the majority of its water from the surface, top 50 centimeters, based on Magnosi's PhD work at KU. And so the more you can partition the limiting resource, which is water in our part of the world, but it might be nutrients or something elsewhere, the better. And so yes, absolutely, would be my answer.
Thanks, Tim. So we do have a couple more questions, and people can even add more if they want in the Q&A section, Tim. So if you could go there when we start the next talk and answer those in written form, that'd be great. Other people will be able to see what your answers were.
You bet.
Great. So everyone will be watching now in the Q&A. And if you're thinking in the next section here, go ahead and submit your written questions even during the talk, and we'll be ready to have questions at the end. I'll pass it over to Tessa.
Great. I'm going to introduce Evelyn Reilly. She is pursuing a master's degree in applied plant sciences in the agronomy and agroecology track of the University of Minnesota. Her thesis work focuses on how intermediate wheatgrass can be used as a strategy to profitably reduce nitrate leaching in Minnesota. Originally from Minneapolis, she finds it meaningful to contribute to the greater economic and environmental sustainability in her state.
Hi, everybody. It's great to be here. I'm excited to share a little bit more about our work with you guys. So let me get my screen shared here. Okay. And let me know if this is showing up okay.
We see the presentation.
Okay,
Yep.
Perfect. All right. So yeah, as Tessa said, I'm a student at the University of Minnesota. I'm pursuing a master's degree in applied plant sciences, focus on agronomy and agroecology. I really like the agroecology portion, looking at that interface between cropping systems and the environment. And so I'll talk a little bit more about our work on nitrate leaching reduction using intermediate wheatgrass. So just to start off with a little background on nitrate leaching in general, Nitrogen is, of course, an essential plant nutrient. It exists in multiple forms. So nitrogen fertilizer can be applied as nitrate. It can also be converted to nitrate from other forms through microbial activity. So for example, it can be applied as urea, and then a series of microbial conversions can occur and it can end up as nitrate. What's unique about nitrate is that it's highly soluble and it's negatively charged. So it does not adsorb to clay particles and it leaches rapidly downward in the soil profile. This is in contrast to ammonium, for example, which is positively charged and will adhere to clay particles which are negatively charged. So there's some unique challenges when we have high levels of nitrate in soil. Total leaching loads are a function of the nitrate concentration in the soil water and the volume of water that infiltrates downward in the soil. So why is this an issue? You've probably heard about nitrate as an issue for hypoxia in the Gulf of Mexico and how nutrient loading can alter that ecosystem balance. In these systems, which are quite far, of course, from the Gulf of Mexico, our concern is more about groundwater contamination. When nitrate gets into groundwater that's used as a drinking water source, it can pose serious human health risks. It can cause blue baby syndrome, among other things. And so it entails major costs to filter and clean that drinking water. It's hard to get nitrate out of water once it's in there. And since many of these are small communities, the cost per household can be quite high to get that water fit for drinking again. In some cases, costing thousands of dollars per household over just a couple years. So how widespread is this issue? It's a serious issue in Minnesota. The EPA limit for nitrate in drinking water is 10 parts per million. 13% of private wells in the state test above 10 parts per million. And in the Central Sand Plains region, that goes up to 30%. So this is really serious. So why does this happen?
It's just hard to measure.
Primarily due to leaching from annual cropping systems. So this data is actually from some work done by the U of M Extension, and it's from tile drainage, so it's a little bit different, but it gives us a proxy indicator of nitrate concentrations in soil water that's a couple feet down, and that's below what we would expect to be the rooting zone. So for corn and soy systems, which are, as you guys know, very common in Minnesota, we're seeing numbers anywhere from 22 to 28 parts per million, so over twice the EPA limit for drinking water. This is really high compared to alfalfa, for example, or Conservation Reserve Program land, where it's 1.6, 0.7 parts per million. That's
hard to measure.
Those are much better numbers to see. So what has Minnesota done in response to this? The state has designated wellhead protection areas. So there's 1.2 million acres total. And these are lands that are considered the source areas for drinking water. So they're areas where water is percolating pretty much directly into aquifers that are then used for drinking. Of these acres, 360,000 are designated highly vulnerable, which means there's a particularly short transit time to the aquifer. Of those highly vulnerable acres, about a third are actually in row crop production right now. So you can see why, based on some of the numbers we have about nitrate levels in water under row crops, this poses a problem. So what are our goals? We want to ensure that Minnesota communities can farm without harming their drinking water quality. We want to provide farmers with cropping system options that are profitable and sustainable. And so the specific piece of this that we're working on is demonstrating Kernza's ability to reduce nitrate leaching into groundwater. So our overall approach to this is we're measuring nitrate concentration in soil water using lysimeters. And if you're not familiar with those, it's basically just a long tube with a porous cap on the end. You can put suction on the tube and it will pull water in at the base. So that can be 2 feet or 4 feet down, whatever you want. And then you can collect water at that depth, analyze it for nitrate concentration. We're also measuring soil water content at 6 depths with a soil moisture probe. With that data, we can model drainage, because once we have soil water content, plant water use data, which already exists, and precipitation data, which we can obviously get, we can figure out how much water is likely moving downward, infiltrating through the soil profile. Then, with the drainage data and soil water nitrate concentration, we can model the total nitrate leaching loads. So here's just another way of visualizing our approach. On the left, we have a lysimeter. So the soil water is collected a couple feet underground in there. And then on the right, we have one of our soil moisture probes. And then using that data, we estimate the nitrate leaching load. So we've used basically the same approach for a lot of projects, some completed, some in progress, some in the planning stages. So some previous work has looked at corn, Kernza, and switchgrass. And this is a valuable comparison because corn is a very different system. It's an annual. We obviously would expect to see different numbers. So it was interesting to compare it to switchgrass, another perennial grass with a deep and extensive root system. So for this experiment, there were moisture sensors at 50 and 100 centimeters, lysimeters at 50 centimeters down. And this resulted in some pretty dramatic results. So there were much lower soil solution nitrate contents under Kernza and switchgrass compared to corn. So on the axis here is soil water nitrate, and corn is the gray line, and you can see that's just dramatically higher through almost the entire growing season until things start to dry out a little bit in the fall. And interestingly, Kernza is even significantly lower than switchgrass in terms of soil water nitrate content. And these are all fertilized at the same rate. So we also looked at soil moisture. And interestingly, Kernza and switchgrass are significantly drier than corn. And so going back to that, the modeling piece about calculating total nitrate leaching loads from drainage and nitrate concentration, if there's less water moving downward through the soil, that means lower nitrate leaching loads. So we think that Kernza may simply be better at taking up water due to its extensive root system, transpiring that water, storing that water, and that may be part of why it's so good at reducing nitrate leaching. So then you can see here, those two pieces were used to calculate the total nitrate leached in kilograms per hectare. So under high fertilizer, really dramatic results, still almost no leaching under Kernza, a little bit under switchgrass, and a lot under corn. And even with no nitrogen fertilizer, there was still significant leaching under corn and then very little under Kernza and switchgrass. And this is really important because we do need to fertilize Kernza to maintain grain yields. And so our data are showing that we can fertilize it without impacting the nitrate leaching reduction benefits at all, which is really exciting. So our current research, building off that initial study, we have 3 sites right now: Staples, where we have a plot-based study, and then we have field scale trials in Pipestone and Chatfield. So here's where those sites are located, and this is the same map I showed you before of wells that are testing high for nitrate. So our research sites were chosen carefully to be in areas representative of the problem areas. So there's a range of precipitation. But interestingly, when I was looking up this data, you can tell that the averages are trending upward. And in the Midwest, we're predicted to have wetter springs as a result of climate change. And so if we're just receiving more water on the landscape, especially in the spring, before row crops are established, there's a higher risk for nitrate leaching. And so having a crop like intermediate wheatgrass on the landscape that's already established, that is already so good at taking up water and taking up nitrate, could be a really valuable part of our climate change adaptation strategy. Another important thing to note here is sandy soil, especially in staples and pipestone. So that means quick infiltration, low water holding capacity in the soil. There's just rapid water movement downward through the soil profile. And here, just to give you a sense of how carefully these sites were chosen, this is an aerial picture showing our pipestone location, and the blue dots are actually existing sampling wells. And then they're outlined in red are our fields. So they are located right there, right on top of the aquifers that are supplying these wells. So some more information on the experimental design. In staples, we have plot trials. So this has been really useful for being able to compare side by side a typical corn soy rotation and Kernza. This is 3 years. We have 2 treatments, 6 reps. So we have, as I said, the corn soy rotation and Kernza. We have 2 lysimeter monitoring stations per plot. And then we have 1 soil moisture point per plot. That measures at 6 depths. And we've had, again, pretty dramatic results from this experiment. So the first year was the soy, and we did see a dip in soil solution nitrate levels under the soy kind of towards the end of the growing season. Things were a little drier. But then post-harvest, they go right back up. And this is unfertilized soy. That's important to note. Whereas with Kernza, that was planted the previous fall. And once that gets established in the spring, you can see that the soil solution nitrate concentrations just drop off and stay down. They never increase after that. And then again, just for reference, the red line is the EPA drinking water limit of 10 parts per million. So Kernza, as soon as it gets going, is just well under that limit, whereas soybean, even without fertilizer, is above it for a lot of the growing season and all of the shoulder seasons. So then the next year was the corn part of the rotation, and you can see that Kernza just right off the bat stays low, whereas corn starts well above the 10 parts per million limit and is concerningly high throughout the growing season. And this was, in this case, this is fertilized corn, but the Kernza is fertilized as well, and we just don't see anything near the numbers we're seeing under corn. We also noticed some interesting trends in soil moisture under Kernza compared to the corn soy system. There was lower moisture under corn at 100 millimeters and 1,000 millimeters at almost all points during the growing season. And here's just a kind of a different way of visualizing our soil moisture data. So along the bottom is actually the soil moisture levels, and along the y-axis is the depth. And then each panel is a different sampling date. So we just wanted to be able to show it all together in a way that made sense. So the things we noticed about this is that we have higher soil moisture under corn at 1 meter at every point throughout the year. We also saw higher soil moisture under corn mid-season. And we think, as I mentioned previously, we think that's because we were getting a fair amount of rain, and Kernza may just be better at taking up water, transpiring it, moving it upward in the soil. We also noticed that water is higher under intermediate wheatgrass at medium depths in the shoulder season. So we're still in the process of analyzing this data. We're going to have another year of it and more from different sites. So we want to look more into that, but it's interesting so far to see significant differences under different cropping systems. We've had similar results in field scale trials. So these are kind of more straightforward. It's just one treatment, big fields of Kernza. The Chatfield and Pipestone fields have been in place for 3 years. We have 4 lysimeter pairs per field and 4 soil moisture access points. And we have gotten really promising results. These are not fertilized, so that part's a little bit different. But here again, you have the red line indicating the EPA drinking water limit for nitrate of 10 parts per million. And then you almost don't even notice it, but the gray line on the bottom is the nitrate concentrations in our soil solution samples from Pipestone. So we had a lot of zeros. I think occasionally we'd have 1 part per million, but it was just incredibly low the whole time. Very similarly in Chatfield, we'd get a lot of just not detected. And what we'd like to do is do some field scale trials with fertilizer, which, based on the plot trials, we expect that we'd have similar results with really dramatically reduced nitrate leaching, but that's one of our next steps is to explore that further. So continuing work, this LCCMR1 round of projects ends in June, so this month. But that's provided 3 seasons of data and a great mix of plot scale and field scale data. We are getting started on the Rochelle Research Farm project. We have plot trials up there with Kernza, alfalfa, and prairie. So that should be a really interesting combination of perennials. And we'll have lysimeters and soil moisture measurements from that. We're also working on establishing the Cold Spring on-farm trials. So those are existing stands of Kernza, and probably in the next week or 2, we're going to be installing lysimeters and soil moisture access tubes. So we'll be able to collect similar data from that. I want to say a big thank you to all of our partners. This work is not possible without all the individuals and organizations and funders that worked with us. So again, huge thank you to them. And I think now we can open it up to questions.
Great, thank you, Evelyn, for that nice talk this morning. Just want to call everyone's attention to the Q&A button. If you want to submit a question, please click on that and type in your question, and we will answer them here live online. If we run out of time, we'll do it by typing back. We do have a couple of questions already, Evelyn, so I'll go ahead with those.
Okay.
Jeremy Schmutz. For the switchgrass corn Kernza combination, what year in the field for the Kernza and the switchgrass were these measurements taken?
Oh, let's see. I'm not sure that if Jake's online, he might be able to type a response to that. This was Jake Younger's my advisor. This was his study, and I'm not sure. I think it might have been 2016. And I do have the study somewhere.
So I'm not sure what Jeremy's asking is if he's asking about the calendar year or the year after establishment. So do you know how old the Kernza was or how old the switchgrass was?
Oh, no, I'm not sure. I'd have to ask Jake about that.
Okay.
This is Jake. I'm online here.
Oh, great.
The study took place over the course of 3 years. And the data that you showed, I believe, were after the first year. And all of the rest of the data are in the AURI Ecosystems and Environment paper that came out last year. But yeah, it was 3 years of data over 4 locations, I believe.
All right, thanks, Jake. Another question, has Kernza been established in a buffer strip at the toe of a hill before surface water and soil water leave to filter the nitrogen.
That's a great idea. And actually, somebody was asking me about that last night. I don't know if anybody has done that, but I feel like there are some locations where that could be, that could work really well, just because it seems to be so good at nitrate uptake. But if anybody else is familiar with use of intermediate wheatgrass and buffer strips, feel free to hop in if you know more about that.
Jake, are you guys planning anything like that?
We've been talking about doing that for years. We want to test it, but we haven't found the right grant or the funding to do it. So something we want to do, just haven't made it happen yet with the funding.
Good, thanks. So what are another question here, what are the tillage practices used in the corn, probably corn soda rotation described?
To my knowledge, I think because there's really small plots, I think they tilled it with like a rototiller, but it was tilled. It's not like a no-till system, if that answers your question.
Yeah. So Jeremy Schmutz asked again, so to be clear, these measurements started the year after establishment, or the switchgrass was planted 1 year and then the data started being taken the next year? Is that right, Jake?
Yep.
Yeah, it's a little bit weird because the Kernza was probably planted in the fall and then the switchgrass is planted the next spring, right?
Yeah, and I don't want to get too into the weeds here, but there were sites where we established everything in the spring, even the Kernza, and then some sites where we established the Kernza in the fall and the others the following spring. And that's all, the details are explained in the paper, and it's kind of complicated.
Good, thank you.
Maybe we can make sure a link to the paper is posted or sent out or something, because that's a great paper that people might be interested in.
Sure, we could stick it in the answer to that question. Another question is, Kernza has, oops, already saw that one. What different nitrogen treatments have been researched? For example, spring versus fall, split application, manure, other sources.
There's actually, we have a trial going on right now. Jake could probably talk more into this, but we have like a nitrogen rate and timing trial going on now. So we should have some interesting data coming up on that. I don't know if there's any other existing data on that, Jake?
That one doesn't have lysimeters beneath it, but yep, it's a nitrogen rate timing trial. And then we also have a bunch of legume intercropping trials going on, and we have plans to install lysimeters beneath some of the legume intercropping trials, the monitor leaching beneath that sort of nitrogen management system. Yeah.
Definitely the older Coleman study included manure versus nitrogen in a conventional versus organic, and the organic leaching was, I think, slightly less, especially in the wheat, but in the Kernza it was almost zero. It didn't matter in the system that you used. So in some ways the timing and placement and things don't matter as much if it's going to be close to zero no matter what you do. And we've kind of done the worst case, over fertilizing with chemical nitrogen fertilizer, and still had pretty low leaching in most of these trials. So I don't see any more other questions coming in. Give anyone a second here to type one in if they want to. Otherwise, I think we're about ready to wrap this one up. Thanks a lot, Evelyn.
Yeah, thank you guys. Great to be here.
We will talk to you later.
All right.
Thanks so much. We're going to shift gears a little bit now and start talking a little bit more about processing flows and then about disease and how that affects processing. But first, just talking about processing flows. And I'm going to introduce Reilly Gordon. He's an engineer for the Agricultural Utilization Research Institute, also known as AURI, which a couple of folks have referenced in talks this week. He primarily headquarters out of AURI's co-products lab in Waseca, but also delves into projects related to AURI's other focus areas of bio-based products, renewable energy, and food. And he holds a bachelor's of science in physics from Brandon University in Manitoba, as well as a degree in civil and environmental engineering from the University of Minnesota. So Reilly, thanks so much for agreeing to present.
All right, good morning, everyone. Thanks, Tessa, for having me, and good morning to the Kernza group. So let's see if I can there we go. So I'm gonna, present a little bit about what we've been learning over at AURI on Kernza processing flows. Just kind of first off a little bit about AURI, just in case some of you aren't familiar with our organization. We've been around the state of Minnesota since the late 80s. We just had our 30th year anniversary. And our sole purpose is to work on value-added agriculture here in the state of Minnesota, so fostering long-term economic benefit through development of new value-added uses for food and agriculture products. We work with small, medium-sized businesses and entrepreneurs all the way up to larger companies and help them develop new products with agriculture. And I guess instead of kind of the more production side that we've heard this morning, we're kind of more on the applied side. So once that crop is harvested, or that animal's harvested, that's where we come in to kind of try to add value to those products. And we work under 4 primary focus areas, as Tessa had mentioned there: co-products, bio-based products, food, and renewable energy. And I do headquarter of the Waseca lab, which we focus more on the co-products, producing new fuels, fertilizers, feeds for different groups based on their waste products, but get pulled into the other 3 areas as well. So. And just kind of showing we have a range of clients all around the state of Minnesota and beyond. We have 4 different locations. So we have the Waseca Co-Products Lab, where we have pilot equipment, pelleting, hammer milling, aspiration, different separation, oil pressing, a number of pieces of equipment, new fluid bed dryer that we just added down there as well. And then over in Marshall, we have our full food lab and analytical chemistry. And then in St. Paul, we have a number of our food scientists kind of just stationed at home, but they've done a lot of work on our Kernza project as well. That I'll share a little bit of that today at the end as well. And then our headquarters is up in Crookston, Minnesota. So just a little bit of a background on AURI and where we're all located. But just wanted to kind of share right away that we're fortunate to have very good partners in some of the work that we've been doing here with Kernza. It's been working with the University of Minnesota and Stearns County Soil and Water Conservation District. They were awarded LCCMR funding through that Environment and Natural Resources Trust Fund. And so we've been written into a couple of grants there to kind of work on some value-added opportunities for Kernza, looking at the processing, looking at different food development strategies, things like this. And so I've been lucky enough to be the principal investigator for AURI on that and kind of leading the effort, but a lot of good partners. We've had Forsberg's, Buehler, Bratney, a number of equipment manufacturers and suppliers really provide a lot of help to understanding some of the process flows and some of the particulars and helping us run trials. NCI, shout out to them for doing a milling analysis for us that'll be included in our report. Montana State University conducted a malting analysis, and working with Sprout Labs as well to, I think you heard Christopher earlier this week, and working with them kind of to help them select some different screen sizes and equipment for them to do some small scale cleaning and dehulling. And then they also manufacture the small malt machines. So that's, they're definitely an interesting partner here right near us. And then Healthy Food Ingredients, definitely have to give them a shout out. And Chris Wiegert in particular, who'll be speaking after this, he's been an amazing contact to have. Was able to do a tour up there, and just amazing what they've been able to accomplish up there. And I've learned a lot from him. And if anyone has any difficult questions for me this morning, I'll probably be passing it over to Chris because I know he's on the line. So just getting right into kind of what we've been learning here on the Kernza cleaning and dehulling process flow. Kind of quite a bit going on here, but I'm going to kind of go through each step for everyone. Just kind of right off the bat, I'd like to say, these are a couple ways that we've found out how to clean and dehull, but there's definitely different ways of doing this. Utilizing, if you're looking into becoming a processor, a cleaner, or a dehuller, you know, looking at if you have existing equipment that you can possibly use and retrofit and modify to do this, it could be possible. So that's something that AURI, you know, feel free to reach out anytime if you're looking into doing this and need some more information. Be happy to kind of share some stuff with you and help you kind of work through that. But there's definitely different ways of doing this. It's just kind of outlines kind of the main steps that we've identified through our research and testing. So there's a couple options, and this is option one, and it kind of I would call this kind of the recycling option that we would go with here. And so taking Kernza straight from the combine and immediately before coming into your facility, you know, testing that grain for moisture, molds, mycotoxins, you know, vomitoxin and aflatoxin, vomitoxin in particular being a common issue, especially this past grow year. Doing a standard plate count, things like that, and make sure you test it in the holes or in the kernel themselves and not the holes, where it is an important point there. But I won't say too much more on that because I know Chris and his group's going to talk a little bit more about that after. So then the first thing you want to do is kind of remove that larger dockage as it comes in. We have a shaker table down in Waseca and a screener, a circular sieve that we used to do that. And then we did some indent separation trials as well with one of the groups that we worked with. And I mean, I'm kind of a little bit reluctant to show actual screen sizes here on anything because a big takeaway of what we learned is you're going to need to maintain flexibility in your setup and have some different screen sizes on hand because the 3 or 4 lots that we handled were quite a bit. There is some variance, significant variance between those that needed us to alter our screen sizes for doing some of this stuff. So that's an important point, to remain flexible in your setup and kind of have some additional screens on site. I think the best way I would think of doing this would be to kind of have several different kind of hand sieves on hand, and a lot comes in, you kind of do it on the tabletop first, and then you would have to make some alterations to your equipment to be able to, you know, run this. But first off, yeah, so once you remove that large dockage, which consists of, you know, just like unwanted straw, biomass, some foreign material, weed seeds, things like that, they get through the holes after there's some free thresh grain in the combine. Then you go to, so you're removing those holes there through that second step. And we found about 800 to 900 CFM on our aspirator, worked pretty well to remove the holes and not pull off any good grain. But that could be, you know, that could vary between different pieces of equipment as well. So then after that, so your holes, you can see those are going to be used as either feed, fuel, or bedding. And we actually characterized the holes for feed, fuel, and bedding that we'll be including in the report as well, just because that's kind of our wheelhouse there, is looking at co-products and where the value could be for that. But I won't share any of that today. And then the grain in hole with whole grain. So this is when it comes in, you're going to have a certain percentage of it that's free threshed in the combine. So separating that off before you go to the dehulling. We found that you know, a 3 to 4 64ths by half-inch slotted screen, somewhere in there, just a good example of the variance. Again, we had one lot that worked pretty well with that 3 64ths, got a majority of that separated out, and then for another lot, it was a 1502 actually was the second lot we tried that didn't go. The 3 64ths just didn't work. It was just too inconsistent. There wasn't making that separation, so we ordered a 4 64ths, and it happened to do the job a lot better. So just an example there of it. You know, taking different lots are going to have different screen sizes, and so this is just kind of a ballpark. And an 8mm indent as well was something that we found that worked pretty well for that separation. And then so after you separate the in-hole from the out-of-hole, the dehull grain would go to a color sorting. All the lots that we saw had at least a little bit of ergot affected grain in it. So that's going to be a necessary step in your operation for sure. And then, so then the grain in-hole goes to dehulling. The dehullers we worked on a few, and depending on the machine, you know, around 1,000 to 1,500 RPM is where we kind of found that to work best with each machine's different, and we found the stone impact ring actually worked the best as well. And then basically what you'd be doing here is just after you go through the huller, you then go through that aspiration step again where you're removing those hulls. You do the separation of your hold from your unhold. Depending on the first pass efficiency of your dehuller, you'll have to do that recycling a few different times. If you have one of the better dehullers, there's a few out there that get, you know, above 90% efficient, which is, if you're going to be operating a large facility and going for that efficiency, then those are kind of the ones you'd like to look for. So yeah, so that's kind of the main process there. And the main point here is you're recycling that to your existing aspiration equipment and your existing separation equipment. And these steps could be done in conjunction. This removal of large jockage, the separation process, and some of the air can be done with like an air screener or a fanning mill type setup. There's certain pieces of equipment out there that we actually tested on that will do that all in one step. But this just kind of breaks it off and just kind of shows what's happening there in individual steps. And there's different ways to achieve it. So, but option 2 would be, and this was one recommended by one of the equipment distributors, instead of doing that, so everything's kind of the same up till that dehulling step. And then instead of recycling over to your aspiration, you actually send off to a separate aspirator. And this way you can kind of keep the process continuous, and rather than offsetting some of your incoming flow with that recycled flow, you're able to kind of remain continuous with it, be a little bit more efficient, and then have that go to a gravity table, which actually has a little bit of a better separation after the dehulling. It's additional equipment, it's additional expense, so you kind of have to, this would be kind of based on, you know, what you're trying to achieve and your budget, but this is just another way to do it. Instead of that recycling, it's kind of going through completely continuous to separate pieces of equipment. Same kind of thing after the gravity table, you're recycling to your dehulling, you're going through aspiration, separation, and separating the 2 streams there, and then everything that's dehulled goes to color sorting, and then you get a pretty nice clean dehulled Kernza grain. So that's generally the process that we identified here. And then just some rough numbers here, and this was a tough one for me to put together as an engineer. Chris calls me the Mr. 100% because I like to have numbers, you know, add up completely, and that's not always the case with Kernza. It's kind of, there's definitely a wide range of some of these steps and what you're going to see come off of them. There's you know, different lots have completely different you know, characteristics. So say, just for example, that this just gives you a rough idea, the Minnesota 2019 average for yield is about 500 pounds to the acre. So looking at, you know, that initial dockage separation, which includes, you know, the unwanted biomass, the foreign seeds, some free thresholds, etc. So that's coming off, you know, you're about 10 to 30% of the total weight there coming off, about 50 to 150 pounds. And so of what's left of that, so 70 to 90% of your original lot there, you're going to have about 10 to 30% of that free threshed. For example, we had one come out of Roseau, a 1502 sample that it appeared to be 90% free threshed. And we talked to the group, and they hadn't put it through any sort of dehulling or anything. It'd been handled a few times, and a lot of it came out. And so just the way it's handled, the combine, the harvest practice, all that can really affect kind of what you're going to be seeing with any particular lot. So just being, you know, prepared to see some different things with different lots is the main point there. But yeah, generally, and we only handled, you know, 3 or 4 lots, but talking to Chris as well and kind of what they see on average up there, you know, 10 to 30%, you're going to have free threshed, and, you know, closer to that, then 70 to 90 by process of elimination, you're going to have that needs to go through that dehuller step. So then pulling off the hulls from there, we did, you know, 5 or 6 different weight tests on the hulls versus the kernel, and we found it kind of in about the range of 25 to 35%. Again, there's definitely some variation there in the hull to kernel ratio, but generally it was about 25 to 35% hull, which would be 65 to 75% kernel as that kind of weight ratio. So just using that, you're going to have, you know, about 60 to 120 pounds of hulls coming off through dehulling. And then you're going to have that free thresh grain that's coming through and meeting that stream of the dehulled stream. coming through. You're going to color sort for your ergot. Just with inconsistencies between, even within batches, you're going to lose some good grain going to like a highly pure product. You're going to have some, dead heads that don't have any grain in the hull itself. You're going to have damage to the grain, maybe through dehulling if you're trying to, kind of hug the line of what the highest RPM. You might have a little bit of damaged grain going through there. You're going to have some general losses throughout the process as well, but that's just kind of real world. So I guess the overall interesting takeaway from this is that you can go from 500 pounds coming in, and generally you're going to see 30 to 50% of that on the back end as clean dehulled grain. We were kind of seeing up kind of between 40 to 50%, but I know that HFI sees closer down to that 30 to 40%. They they're going to a 99.9% pure product, and so there's just some additional losses that come with that. So yeah. And here's just a couple pieces of the equipment that we worked on from our trials. Up top right here, this is a Forsberg friction dehuller, and then in the middle, this is a Forsberg impact huller. Working through both the friction and the impact, we definitely found that impact is the way to go on this, just a little bit higher efficiency, just a cleaner dehull all around. And then here on the left is just showing you, I think this is a 1/18 by 3/4 oblong, and just showing you like this was a pretty close for this sample, but as you can see, there's some of the grain that gets stuck in there, and some of that is still in hull and some of it's not in hull. So there's some grain inconsistencies in each lot. It's a pretty fine line of what screen's going to work the best there that we found. So having several screens on hand to kind of swap out for different lots is going to be important. And then you can see down here, this is an indent cylinder separator that we use. And then the Kais aspirator is the one that AURI has. We actually, AURI under our second LCCMAR funded grant with the Stearns County Soil and Water Conservation District, we've had some equipment dollars, so we've actually purchased this Forsberg dehuller and this indent cylinder separator from Bratney to be able to do, just some small scale cleaning and dehulling at our Waseca location. We plan to get that certified as food grade. We have a newer location that's, it's not going to be in with all the dirty stuff in our co-products lab, but we kind of have another building that we've rented out for our fluid bed dryer that'll remain quite a bit cleaner. So we're hoping to get that certified and bring our aspirator over and do some small scale cleaning and dehulling to support some different pilot projects here in the States. We're excited to be having that as a capability. And then just here in the middle, this is a fanning mill that shows you just kind of the multi-floor setup that I mentioned, where you can kind of do the larger dockage removal on the top and kind of work on that in-hole from out-of-hole separation near the bottom, with air kind of moving through consistently. So you're getting kind of a number of steps, killing a couple birds with one stone there. And then just up here is just finally a picture of a color sorter that's on the market. That's another piece in this operation. And so just here is just a little bit of a teaser for some of the stuff that we'll be releasing here as part of our first report with the University of Minnesota. That project's set to wrap up at the end of the month. So we're working diligently now to get that all wrapped up. And so here's just a couple of the one-pagers that we'll be releasing kind of to pass out to different, potential partners and interested parties. So we have the one on the cleaning and dehulling that I kind of went over today. Our food team did a great job putting together a few different ones on our analytical testing that we did on the grain for different food applications. So what that looks like compared to wheat for baking, what it looks like for brewing. We just have a general cereal grain one pager as well that we're excited to be able to have those ready to pass out to folks. And the report, in addition to this cleaning and dehulling research that I went over today, it's going to contain, sections on recommended potential food uses from those analytical results that we got, our full malting analysis that Montana State University conducted, storage and handling recommendations for the grain. We did some research on guidelines for registering as a food facility, particularly in Minnesota. and what that all entails. And then milling recommendations, that'll all come from the Northern Crops Institute. They did a number of milling analysis for us that we'll be scaling up actually in our second project with some additional funding. And then just an initial co-product evaluation of those straws and hulls, and kind of where we kind of see that value being added on those components. And then we'll be looking to also expand that evaluation into actual feed, fuel, and bedding valuations in our second one with the Stearns County project. So next steps for AURI, as I mentioned, we're going to be doing food product development testing, most likely with the Northern Crops Institute. On our list of things to do: pasta, crackers, puffed grain, flaking, breads, pizza crust, tortillas, milk, etc. We're not going to be able to have budget to do all those, so we're going to have to prioritize kind of our favorites there and pick what we're going to do. But we have all that planned out to do some different food product development testing with our food team. We hope to support as many pilot projects and provide technical support for Minnesota businesses as much as possible. So that would involve, helping out with cleaning and dehulling setups, and also working with small to medium-sized businesses to give some food science recommendations and helping out with doing some of that work. And then we'll be doing expanded co-product testing. As I mentioned, we plan to do just kind of an interesting one. We looked at the lignin, hemicellulose, and cellulose content of the straw, and found out that it was, we connected with Chad Olvin at C2 Renew in Fargo, one of our contacts there, and he's thinking it's going to work pretty interestingly for a biocomposite. So we're going to have some mixed with a PLA resin. We're going to use the Kernza straw to make a biocomposite. Maybe they've been able to do some 3D printing with biocomposite material, maybe 3D print a beer stein and a cereal bowl and some utensils and things like that, just to kind of keep it Kernza related and kind of show another interesting use of those co-products. And then we'll be doing the bedding analysis, fuel and feed valuations, as I mentioned as well. And then just continued outreach and dissemination through our AURI Connects team. We have a new event called Fields of Innovation, which at least one of those events is going to be Kernza focused, where we're going to have a number of presenters and networking around Kernza, and hopefully that can take place, in person when this COVID stuff blows over. So other than that, I think that's all I had for you guys today. Really, thanks for having me again, and any questions, regarding more information on Kernza, being a pilot project partner here in Minnesota, feel free to reach out if you're interested, and thanks again. So.
Hey, Reilly, thank you very much for that presentation. That was excellent. I imagine there will be some questions coming up. So anyone who has a question, please go to that Q&A button and type in a question, and we'll take them here and talk about them. We have plenty of time to do that. So we had one question already about whether your slides are available, and Tessa said that you did already give her a copy of those that we can share it with people. So
absolutely, yep.
That's great. Just want to remind all the speakers, if you can give us permission, we asked you for that already, but to share your information, contact information, and your talk today, record it. Perhaps you've already all done that, but we're planning to post these all online. We'll get those links sent out when we've got them posted. So should be coming up soon that we can view these and share them with people who might have missed. Yeah, still waiting on any questions here, but I've got one to start with. Thanks for showing all those pictures of that equipment. That's really cool. The dehulling that you did, you mentioned the RPM. When you crank it up pretty high, you starting to break up the seed pretty badly. I mean, can you drive it to that point? Is that kind of what happens?
Correct. Yeah, that's kind of the trade-off there, is you're either too low and you're not dehulling enough, you're not efficient enough. You're trying to kind of maximize that first pass efficiency to therefore maximize your whole process flow. But once you start turning it up high, there's a fine line there of starting to have some breakage in the kernels, and that's obviously something you don't want. And again, that's going to likely vary between different lots. What is interesting is that the University of Minnesota is working on, the genetics there and coming to a more of a free threshing variety. And at that point, then, dehulling may become less of a necessary process step, which is kind of interesting to think about.
Yeah. When you were getting to the end of your process, what percentage were still in hull? Are you at almost 100%, or are you allowing a few percentage of in hull seed?
Yeah, so different dehullers have different efficiencies. So the one there's a one that's called the Shuli that's was over 90% efficient that we tested on, or around 90%, and then the Forsberg ones were definitely less efficient, somewhere around 50% on that first pass. And it really comes down to the Forsberg's also quite a bit of a cheaper piece of equipment as well. So that's why really we went with that, because we're not in for production. We're just going to do small amounts, and we can afford having to recycle a few times. But if you're in production, that's kind of where you want to understand you want to get that greater efficiency machine. So it really depends on the machine. We kind of saw it all over, and I'll be documenting those in our report as well, and kind of what first pass efficiencies we did see on those different machines.
What kind of throughput those machines have?
So the Forsberg one that we are getting will have a couple 100 pounds an hour. The ones like the Shuli, a Bueller, a Kodima, are a couple other ones that we have on our radar, and those are closer to 3,000, 3,500 pounds an hour. So depending on what your throughput and what your needs are, you can find different pieces of equipment that can fit that. So
good. We've got a few questions here. Of all the things a farmer can do prior to and after harvest, what do you think farmers should focus on to improve the grain quality, harvest timing, combine aggressiveness, drying conditions, etc.?
I think all those are important. I think drying conditions and techniques are something that we haven't looked into yet. And I don't know, that's Jake asking the question. I think we might be actually planning on looking into that with Jake under a new Kernza grant that we're focusing on. But I think, combine aggressiveness, if that's implying that, you're going to have more free thresh grain in it, that just kind of takes out an extra step. And that's, just more efficient through the process on the back end. I think also, moisture, I think always drying conditions, moisture is always the biggest issue, because if you have mold issues, you don't really have a usable crop. So I think that's number one for anything in my mind.
Yeah, something I've noticed, I don't know if all farmers would share this experience, but we've been, by using a traditional combine instead of the modern combine design, we were able to get probably more aggressive dehulling in the combine, kind of cleaner seed than with the modern rotary combines. Actually are a little gentler, and therefore it's hard to really get that aggressive dehulling. Sometimes if you find a really good sample, it might be someone who had an older combine.
Yeah,
it's been easier for me to get that. Question from Carmen, is storage impacting, I think, post-processing like dehulling? I think he's asking about the impact of storage on dehulling.
Oh, like storing dehulled versus storing in hull?
I'm not quite clear on what he's asking. Maybe Carmen can clarify that for us. But the storage impacted post-processing like dehulling.
I think, if he's asking I think the storage by dehulling, I think you're probably going to maybe have a little bit of storage reduction in shelf life, but potentially for sure if you mill it, you're going to have a reduction in shelf life, but we actually didn't look into that. So I'm not sure. I'd just be guessing there. Maybe someone like Chris, if Chris is on the line, he might have an answer to that one.
That was the question I was wondering about. Does dehulling impact the quality of the grain for storage afterwards?
Right. And from my experience with it, we didn't hold on to it for any extended period of time, like over a year or anything. So it would be tough for me to know that. That wasn't something we particularly looked into, but I think it would be something that would merit some research for sure.
I know that like in oats, if they've been dehulled, the oils aren't stable. You can get rancidity developing after dehulling. I'm not sure if anyone's tested that yet. Certainly after milling, we would have that issue. So
yeah, milling for sure, you're gonna have a reduction in shelf life, and we touched on that, but we didn't look at dehulled versus in-hull. So
yeah. Right. See if there's any other questions. If not, we will move on to our last talk for the day. All right, thank you very much, Reilly.
all right, thanks a lot. Yep,
Thanks for all this great work you did.
appreciate it.
Hi all. I want to mention that Chris Wiegert actually was not able to join today. Today was, or this week was their first week back in the office during COVID, and so he was got taken away from us today. He thought he was going to be able to make it, and then at the last minute had to cancel. But in the meantime, we still have Kathryn Turner, who is going to present on disease and its potential effects on harvest and processing. Kathryn is the crop protection geneticist at the Land Institute. She attended Austin College as an undergraduate and received a PhD in plant breeding and genetics from the University of Minnesota. She was hired as a postdoc at the Land Institute with the perennial wheat breeding program and has been in her current position for 2 years, working to understand, identify, and control diseases in perennial grain crops.
All right, thanks, Tessa. Just getting my presentation pulled up here. Jumping in with ergot. So ergot is caused by Claviceps purpurea. It's a fungus, and the symptoms include gangrene and hallucinations. It overwinters as fungal bodies in the soil or on the surface of the soil, or in crop residue. So the FDA limits toxins in grain, and for ergot, the limit is based on the proportion of the fruiting bodies, the sclerotia, that are in the grain by weight. So, and it varies by different crop. It's 0.05% for wheat, and then slightly more for barley, oats, and the highest level for percent rye. And that's based on the amount of grain that's consumed. So for a diet that includes a lot of wheat, the level is more strict. In intermediate wheatgrass, we're currently not quantifying the amount of ergot, and that's because it is possible to remove it in some of the processing steps, but it's present in all grain lots, and that's based on observations from HFI. And the analysis that they're doing. At the Land Institute, we typically see kind of low levels. So we have a disease nursery, and in 2019, looking at 5 heads from each of the 500 plants, we only found 4 that had ergot bodies. 2018, very similar levels, and the amount of toxin that was in those plants that were infected was about 4% of the total weight of the seed. And so there are issues with cleaning the ergot out. When we clean the grain, we can get rid of the ergot, but that results in a reduction of yield because some of the grain is also lost. So cleaning a batch that might have 5% infection could result in about 10% loss of total yield. Another fungus that causes toxin is Aspergillus species. There are multiple species. The common ones are Flavus and Parasiticus, and they produce aflatoxin in the grain. This is a saprotrophic fungus, so it feeds on dead, decaying material, and it's present in most soils. It causes cancer in mammals, and that's why it's regulated. So the FDA is pretty strict. They don't allow any mixing of lots that exceeds thresholds, and have very strict laws in place to prevent that. The limit is 20 parts per billion in food and a little bit higher, 100 to 300 parts per billion for livestock feed. In wheatgrass, we're seeing pretty low levels. HFI is testing for this routinely with all of the grain lots that they get. They haven't seen anything as high as 10 parts per billion. So it's something that we're continuing to monitor. And in some years it could be an issue and for some grain lots. So it's something that always has to be checked. But this particular toxin is, it tends to be more of a problem in corn, and mostly during the harvest and storage and transportation, particularly if there's really high temperatures or excessive moisture. And for this one, you can often see the head will be kind of black or have a moldy appearance. And third, we have DON toxin, which is also known as vomit toxin. That's because it can induce vomiting and feed refusal, loss of weight in livestock. Humans, causes hemorrhage and shock. It's commonly produced by multiple species of Fusarium and causes a head blight in the cereal grains. So there are multiple species that produce this toxin, and then some of those species can also produce other toxins, but DON is by far the most common. The FDA limits consumption levels for humans to less than 1 part per million in the final product, 5 parts per million for swine, and 10 parts per million for cattle and chicken. And that's based on the proportion of the diet. So at a lower level of the diet, if the grain exceeds those or is at those higher thresholds. In intermediate wheatgrass, we saw some levels above 1 part per million in 2015. And then 2019 was a particularly bad year, not just for wheatgrass, but for wheat as well, particularly in the upper Midwest. And we saw some levels up to 10 parts per million. That wasn't in all grain batches, but there were a lot that were kind of in the 2 to 5 parts per million range. And at those high levels, the grain's not safe for human consumption. And it really doesn't have very much value for humans above 1 part per million, and for animal feed, much above 5 parts per million. The processing facilities typically won't handle grain that has more than 2 to 3 parts per million. And that's partially because of the inability to reduce the toxin levels to a large extent. And then there's also a lot of expense and time in cleaning the equipment and reduced grain yield as well. So one kind of tricky thing about the DON toxin accumulation is that it can occur when there's really low level of grain infection or disease infection. So we survey diseases of intermediate wheatgrass regularly every year. And Fusarium head blight, in terms of the number of plants that are infected and the severity in terms of the amount of the head that is infected with Fusarium, is really low, especially compared to other diseases. But we still can see pretty high levels of DON toxin. So in our irrigated inoculated nursery, there's a range of 0.5 parts, or the average was 0.5 parts per million, and we saw levels up to 5.6 parts per million. And another kind of difficult thing is it's not always possible to determine which grain is infected. So on the bottom, these are somewhat infected. There might be a little bit of an orangish color here, and these are a little bit black in color, but they don't look substantially different from the uninfected kernels. And you can't see even this part until the grain is threshed out. So where does Fusarium come from? For other crops, this has been studied pretty well. a substantial amount of the inoculum comes from within the field. And that's about 30% of the inoculum in wheat. Then there's a larger component that's probably coming from the regional level of spores produced and from atmospheric sources. So the majority is coming from outside the field. There's some practices that increase the production of fusarium. Corn has pretty high levels of fusarium, and planting into corn stubble can be a problem for wheat and probably also for intermediate wheatgrass. And then not tilling is also produces higher levels of infection. So for wheatgrass, yeah, burying the residue through tillage might be a kind of a stopgap measure. It sounds like that from Jake's data might also have a potential to increase grain yields. It's not something that we want to do long term, but in the meantime, until we get really highly resistant varieties, tilling between rows might be an option to reduce disease levels. So the spores from within the field come from debris that's left on the ground. There, you can see parathecia, which are the fruiting bodies for the ascospores, which are the sexual spores of the fusarium fungus. So on the crop residue, these appear as little, very small black dots. And in the spring, when temperatures warm up and moisture occurs through rain, the parathecia forcibly discharge spores up onto flowering plants. The other source is from conidia, which are asexual spores that occur in the soil and also on crop debris. Those are typically rain splashed up onto the leaves of the plant, and then over the course of the season are splashed up onto the heads. And the infection occurs when in the grain, when the heads are colonized, the anthers provide very good levels of nutrients and allow the fungus to spread. With wheatgrass, the spikelets are open for an extended period of time during flowering, and we think that this may be a source of infection. So there are a number of practices that can affect the accumulation of toxins. Some of those occur at the field level, so there are biological factors, agricultural practices, things like tillage, residue management, previous crop, and as well as fungicides. And then there is also selection of a resistant variety, and it's also dependent on the fungal species. So there's some strains and species that are more aggressive than other pathogens. But for the disease to be present, the environmental factors have to be conducive. So for fusarium to occur, it really thrives under moist conditions and when temperatures are between 75 and 80 degrees. But without those environmental conditions at the right time of the plant's flowering, we wouldn't necessarily see high disease levels, which is why in a lot of years, the disease levels and toxin produced as a result of the disease is still pretty low. And then there are practices at harvest that can impact the disease and toxin accumulation also. Things that we think might be important with Kernza are swathing or direct combining. So for swathing and laying the heads on the ground, if there is a rain event or something that delays the collection and threshing of the grain, that can increase the amount of toxin that ends up being in the grain. and maybe using different techniques to thresh so that we can remove more holes. So Lee was just mentioning different combine strategies that might help get a little bit cleaner product and reduce the amount of inoculum that's right there with the grain. And then there's the possibility that toxins can continue to accumulate over storage. So if there's fungus still living and there's high moisture above 22% and good temperatures, the fusarium or other fungi can continue to produce additional toxin. And then as we go to malting, there are opportunities in that process also through steeping and germination where there can be increases in toxin levels. There's also some points in the process where you can decrease the toxin levels, and that's largely through extra processing to reduce toxins. So there can be cleaning done to remove dust and debris, and also the holes. And these are, yeah, the parts of the process that Reilly was discussing. We can also clean to remove the infected kernels. So that could be either through wind or gravity. These are kind of challenging in Kernza because the size of the grain is small. And with wheat, we see a lot of kind of empty tombstone kernels that are lighter in weight than the non-infected kernels. But in Kernza, the kernels are really small anyway, and we may see some that have a lighter color, like this kernel on the bottom right. But for the most part, they look very similar to the healthy kernels. So I think color sorting is probably a better option for something like ergot. It might not be as effective for sorting out Kernza. Some processors use blending to mix grain lots that have lower toxin levels and higher toxin levels, but that's really up to the processor, and that's not a practice that HFI uses. And finally, by milling, could expect that, or based on some other research in other crops, that there's higher toxin in the outer layers of the grain, like the bran, and so removing that could reduce the total amount. So the testing processes at HFI, they typically take, or for every grain lot that comes in, they sample at the field. So they have the growers collect a sample from the combine and do kind of a pre-screening to make sure that it doesn't exceed toxin levels. And then they also sample at the gate, and that's using a probe and multiple samples within the same grain bin. And they're currently providing any rejected grain to TLI for research purposes to determine where the toxin is located in the grain. And they don't, as a practice, blend grain lots that have thresholds that are above what they accept. So on the kind of breeding and resistance side of things, we want to try to reduce the possibility of disease and toxin before we get to the point of managing it in the field or having to take it out at the processing step. So the best thing and the first thing that we can do is to select for better germplasm that is resistant to fusarium head blight. So we've been working on this project for the last 2 years. We are employing genomic selection. So the fusarium head blight is a very expensive and time-consuming trait to measure. And by just phenotyping and genotyping a small number of plants, we can use that assessment to predict in a larger population of 10,000 plants, which plants are resistant and which plants are susceptible. So this is work that's done with collaboration with Kansas State University and Jared Crane, who's in Jesse Poland's lab. So we've been selecting 500 plants to put in the field, genotyping and phenotyping them, and then creating a model that we use to predict the resistance and susceptibility in the 10,000 plants that go through the breeding pipeline every year. And for the first 2 years, we've had really high selection accuracies, between 0.7 and 0.9 for fusarium head blight severity and incidence, which is good within the year, but we are having a problem making predictions between years. So the correlations both in the phenotypes and for our models are really low between 2018 and 2019. So there's a few things that are probably causing that. One is high genotype by environment interaction. Fusarium is very highly variable, and without having a replication in the field, we're not really able to get a good assessment of all the genotypes, or of the get a good assessment for a particular genotype. And so one part of the field is likely to be more infected than another, and just the timing of the heads flowering. Our infection levels are also pretty low. Even in our inoculated irrigated nursery, we have about 14% of our plants above the allowable threshold. So most of the plants in the population are fairly resistant, but we may need to increase our disease levels to be able to detect those. So we're confident that even if we're predicting within the year, we can still eliminate the susceptible genotypes within that particular year, which is still useful, whether the plant is susceptible in 2018 or 2019. Those are genotypes that we wouldn't want in the breeding population. And I think the year variation is largely due to having that one plant in the field not being infected at exactly the right time in a particular year. So the flowering time and the availability of inoculum would have to be perfect. And there's a lot of variability within infection for fusarium head blight. So we're planning to increase the number of environments, and then also replicate alleles and genotypes. We also want to start some on-farm research, because we're getting a lot lower levels of infection in our disease nurseries than farmers are seeing in some cases in fields. And so we've proposed some research to start with Kansas State University, Jess Rupp and Myron Bruce, as well as help from George Anon for on the milling. And our plan is to sample fusarium in farmers' fields across the US to identify the species and strains that are in intermediate wheatgrass. So we've collected strains from wheat in the past, and we know that they can infect intermediate wheatgrass, but we don't know that those are the ones that are actually causing problems in farmers' fields, or are the most aggressive for intermediate wheatgrass. We also plan to test grain harvest, cleaning, and milling methods to reduce the toxin content of the grain. So specifically in the research fields, that would involve comparing direct combining with swathing, and storage moisture and timing of harvest for dawn concentration. And on the farm, there were some ideas from farmers who wanted to test differences in stand ages, and the impact of intercropping with a legume. So there's some published research that shows that intercropping with or not intercropping, but having additional biomass in the field, and the study, it was actually weed biomass, reduced the dispersal of spores and was effective in reducing the amount of disease that occurred. And finally, we're going to look at the DON levels in the grain and see if there are differences in but across many different grain lots after they've been dehulled or milled by removing the bran. So with that, I'll acknowledge people at the Land Institute who have helped with this work, at Kansas State, for Chris Wiegert for his comments, and some of the data that's presented here is based on the data that's from HFI, and his input in coming up with the new proposal. And then funding sources, the Perennial Agriculture Project through the Mullen Foundation. All right, thanks very much. I'll stop sharing my screen and can take any questions.
Great, thanks, Kathryn. Yes, if anyone wants to submit a question, just hit the Q&A button and type in your question. We've got a couple minutes here. I'll start out with a question for you, Kathryn. You talked about the aflatoxin effect, which doesn't seem to be very severe, but can you see a seed that's infected with that, or does it not look obvious?
I think you can probably see the spores, the mold on the spikelets and on the head. So it would look like kind of a black mold growing on the head. I'm not sure if you would see any difference in the grain or not. Okay. That's, yeah, something that Chris might be able to address.
Another question that's coming in now. Carmen asks, has work been done in relation to the second and third year fields, I guess, whether it's getting worse over time?
Right. So we have pretty low levels of disease in Salina, and we have only had the inoculated irrigated nursery for 2 years. In 2019, it was not more severe than it was in 2018, but I think the environment plays a huge role in the development of disease. So I don't think it'll necessarily be worse in subsequent years, at least based on our 2 years of data.
Another question, have you found any candidate genes for Fusarium head blight resistance?
No, that's something that we will be working on as a part of the FFAR project that will start in the fall.
Carmen asked, would there be an advantage in removing the residue, maybe burning versus baling, or assuming in your experimental plots, you're also removing the residue?
Yeah, we are baling the residue in our experimental plots. Yeah, I think there might be some reduction in at least the level of inoculum. I think if the field is grown in an area where there's a lot of other wheat or corn or sources of other inoculum, probably 50% or more of that inoculum is coming from outside the field. So there may still be plenty to generate disease levels that are high.
Matt Baker asks, do you know whether Fusarium can spread between spikelets, as in wheat, or whether each disease spikelet represents a separate infection, as is in barley?
Right, so we do know that it can spread between spikelets. There's, before we did field testing, we inoculated individual spikelets in the greenhouse and measured the spread of infection. So just inoculating one spikelet, the disease spreads